here we go. What shape has the maximum ratio surface area to volume? What kind of shape would it be? Well, that's actually maximizing volume the bigger you get. So really, this isn't necessarily the best way. If you're going to maximize absorption, you really want a flatter and flatter shape. So again, there's some kind of a trade-off going on here. What is the advantage for yeast versus long, thin hyphae? Could be something with the amount of oxygen that's present. So, what do you know about yeasts and oxygen? They can be anaerobic for some significant amount of time. So, aerobic versus anaerobic. This is a big thing in life as well. Most eukaryotes are aerobic organisms. Aerobic organisms. Yeast can sometimes be anaerobic for long periods of time. This is rather unusual. So what do yeasts do when they're functioning anaerobically? Fermentation. What do they produce as a product of fermentation? Not lactic acid.
Alcohol?
No. Alcohol. Ethanol. This is where all the ethanol in the world comes from. It comes from yeast. So these are organisms that are in some ways limited by their body plan, but they might have an advantage in certain environments. Maybe it's an advantage in an anaerobic environment to just be single-celled. You don't have to support as much of a mass. Right? If you're a single-celled organism rather than a multicellular organism, what kinds of evolutionary traits can you do away with? Think about the multicellularity, the steps towards multicellularity. What is needed to maintain a complex multicellular body? What's that? Sexual reproduction. How does that relate between single-celled organisms and multicellular organisms? They can reproduce by budding, but hyphae can reproduce asexually as well by dividing mitotically. They can still reproduce sexually as well. Adhesion, communication, no specialized cells for reproduction. That's one of the differences between single-celled and multicellular organisms. So they do away with a lot of specialization. They don't need to have cellular adhesion molecules. They don't need to have all of the complex purification systems. There are advantages because you can get away with not having a lot of the features of complex multicellularity. But again, these are going to be limited to some environments. They may not be competitive in all environments. Oftentimes where you find yeast are places where only microorganisms have a chance to survive. So the kinds of environments where you find yeast are the same environments where bacteria will dominate.
I feel like you've hit a lot of that in that last one.
Yeah.
So fermentation, I think that's supposed to be plants, carbohydrates, sorry, and then leavening bread and alcoholic beverages. Okay, so let's talk about decomposition. So we've already mentioned that fungi are responsible for decomposition. They convert dead organic matter back to CO2 and water. So remember, I mentioned at the beginning that they're very important to cycling carbon. So let's see. They can gain nutrients, remember, from the environment. So that means dead animals, protozoan or bacterial cells, but the most abundant molecules on and within the soils are going to be cellulose and lignin. So that would make sense, right? If they're in the soil, then the plant matter that is there is decomposing and then providing nutrients for them.
I have a follow-up question on that. Why are cellulose and lignin so abundant in soils?
I'm sorry.
Well, why are they so
Nothing.
abundant?
Okay.
But so plants aren't just made of cellulose and lignin, are they? So why are the cellulose and lignin so abundant? Because nothing else can eat it. Yeah, it's hard for other things to digest it. You eat plant matter, you're mostly wasting your time. Because you're eating a bunch of stuff that you can't really digest. There are parts of the plant, the simple sugars, the starches, those things you can break down, oils, but a lot of the structure of the plant we can't digest. And this is true for other animals as well. so that other stuff will build up, the cellulose and the lignin. This is a big, important feature of what fungi do. They can make use of food that other things can't. What is cellulose really? Carbohydrate. What is it made of? Sugars. A bunch of sugars linked together. So everything on this planet can eat sugar. Fungi can eat sugar. If you put yeast in a bottle with simple sugars, they will grow happily. And if you grow them in an environment with lots of oxygen, they will bubble away, produce CO2 from aerobic respiration, just like every other thing that can do aerobic respiration. If you bottle them up so that there's no more oxygen that can get in, they'll produce a little bit of CO2 and then a bit of alcohol. But if you put them in an environment where there are no simple sugars, what can they do? Well, this is something that fungi can do that other things can't do. They can start digesting cellulose. They can even digest wood. Because in essence, all of those things can be broken down into sugars, if you know how. Fungi know how, other things don't.
So I thought these were kind of cool pictures. They're showing you here what regular cedar wood looks like, and then what cedar wood in, let's see, somebody's King Midas's tomb looks like. So the wood was essentially infiltrated by fungi, and so you can see that it's decomposing the wood of that tomb. So I thought that's kind of an interesting comparison, where you can see what it normally looks like versus what it looks like when the fungi are breaking it down. And then in terms of fungi in infections in living tissues, so if anybody here gardens, does anybody garden? Sometimes you see, like, your plants can get different infections and whatnot. And we've had a heck of a time with certain things that we try to grow. But so what you can see in this picture here is that there's a vascular wilt fungus that's actually in the xylem vessel of an elm tree. So you can see that hyphae is basically infiltrated those tissues. And so the other thing you should know is that unfortunately, plant infections can be transmitted relatively easily. So the fungal spores can be transmitted, whether it's by wind or whether an insect lands on something and then picks up the spores and takes it somewhere else. And then below ground, as in this picture here, you can see that the hyphae can penetrate the roots of the plants. Some fungi will enter through the stomata, and there's actually, we're gonna, I think in your slides, like when we get later into this, you'll see what else hyphae can penetrate. So it's pretty amazing. Is that just sheer force that it's able to penetrate? How does it get in there? Is it opportunity? Yeah.
The hyphae are open. I mean, not the hyphae, the stomata are open for part of the time. The hyphae are very thin. So
you can just get in
if
there?
the opening is there, they start growing.
That's fascinating. This is a slide that blew my children's minds the other day. They happened to be walking by when I was prepping. So this is actually a worm being trapped by hyphal wings. So, again, like these organisms are kind of amazing with the way that they can grow and what they can grab onto. And in terms of infections in vertebrates, luckily fungal infections are rare, and in a lot of cases they're usually not life-threatening. I know some people, one of my girlfriends had something called candida. I don't know much about it, but she had like some kind of rash for a while. So usually you hear about it in terms of like athlete's foot, yeast infections.
Right.
So yeah, she had something.
Is she pregnant?
No, she
Is
was
her immune system compromised?
probably.
Yeah. So the candida yeast, they grow on our skin, but they're not in abundance because they're in competition with other microorganisms. But sometimes things get a little out of balance and get infections. It's a common thing in people that are immune compromised.
She had to alter what she did to get rid of it.
Well, some issues do that.
Tell her what she did.
Well, they get a little bit of information and they think, well, I'm going to do the candida diet.
But there is, I think there is a
Yeah, there's a candida diet, yes. Yeah, I read about that. But, no, the infection is usually the result of some, like a major change in your personal environment caused by maybe a drug that you're taking or your immune system being compromised, because those yeast They normally grow on your skin in a certain proportion with other microorganisms, and they wouldn't infect other parts of your body. So when it affects your throat, it's called thrush. That can happen to babies too, but.
Well, I was always nervous with the babies, because if you get it and you're nursing your baby, then they can get it also, and so, yeah, I was terrified. Not that I think it'd be like that big, I don't know if it's that big of a deal, but I was terrified with it.
It's, I mean, I've never had it myself, but
Something I did not want to deal with.
unpleasant.
Yeah.
Athletes flipping
Do you want to mention your amphibians here?
Yeah, but we'll bring them up later.
Okay.
One general idea about this is you have it up here on the slide.
If you have it up there.
Why these fungal infections don't typically cause us a lot of problems is that the fungi really prefer things a little bit cool. They don't like things to be really hot, and they don't like drying out at all. So when the environment favors fungal growth is when temperatures are a little bit lower and the environment is moist. I think that's the word here. Moist. So this also has a lot to do with their decomposition abilities, because there have been times in the past when fungi haven't been doing their job of decomposing wood and plant matter. And it has a lot to do with what's been happening in the environment. When temperatures are a little bit cooler, but not freezing, and the environment is moist, not too dry, then fungi can break down the wood. When things get too hot, fungi don't do a very good job. When things are too dry, fungi don't do a very good job. And that's when the wood and plant matter can pile up. And that leads to the fossil fuels, eventually. Everything that we dig up and set on fire, whether we're talking about coal, oil, even pockets of natural gas, part of that has to do with times in the distant past when fungi weren't as abundant, weren't as active for one reason or another. Oftentimes because of warmer temperatures, or sometimes it's too much acid in the soil or too much sulfur. But these are things that interfere with fungi. That interferes with decomposition, and that means more CO2 has been pulled out of the atmosphere and is stored in solids.
Did you ever have your classes create test questions?
Do I have them create test questions?
I've got 3 at least, just from
That's
like
an
your
interesting
talking.
idea.
And so what I'm wondering is,
I never thought that. No.
you could do it as an exercise
You could do
though.
that. You do it right now. You
Yeah.
tell them that.
Okay.
Bring them
Well,
that.
I wanted to run it by you before.
Why?
So today, let's do this. Like as we're continuing here, if you're hearing things that you're like, because I'm writing things down where I'm like, that's a great test question. That's a great test question. I have, like I said, no, I got 4. So as you're hearing things, if you hear something that you think that would be a good test question, write that question down. Because what I've heard some people do is they'll have their classes like suggest questions, and then they actually will use them.
That means less work for us.
Yeah, everybody wins. And so what we can do is if you write something down that you want to submit, then we can actually ask them next week.
Sure.
And we can review that way.
You even get a little credit for it.
I like it.
If you give a number to it, they'll think it's meaningful. But the
Think
number will be
meaningful.
so small that it's not really mathematically meaningful. Because they're not paying close attention, they will remember that I said this right in front of them. And then they'll do it for that extra credit. That's
I like
how you
it.
manipulate minds and numbers.
So yeah, so and so we can do it this way. We have 30 minutes left. So is that enough time for them to do one today? That way everybody can get in on the extra
Before we're
credit.
done, yeah, we
Okay.
can do that.
So if you're hearing something that you think would be a good test question, write it down. And no joke, I mean, we will definitely look at them and we'll review that way then on Monday and potentially use it on next quiz. When did you want to do that?
I think we gave a quiz on Thursday or Tuesday. What day is this? We're Wednesday. This is Wednesday. So we did a quiz Wednesday. I think we could do another one. I mean, we're going to cover like 3 or 4 chapters in the next week. So either Wednesday of next week or Monday of the following.
I like the polymer. Yeah,
I'll have one.
Instead of next Wednesday.
We gotta get these quizzes going. Come on, work with
We
it.
gotta get going. We're gonna get aggressive on the quizzes. Submit test questions. Put your name on it.
Yeah.
Cool.
Not this Monday.
Yeah, not the 14th. Next quiz, 21st. So, like I said, if you're hearing something, write it down. Looking for test
I
questions.
think it's good now if you just give us a quiz question that you found on the internet or something we've seen before. Come up with something on your own.
Okay. Cool. Now we're getting to the fun stuff. So, mutualistic relationships. So here, we've talked about mycorrhizae before. And we also talked about the fact that there are two main types. There's ecto and endo. So they are what they sound like here. So the ectomycorrhizal fungi, sorry, I like fungi. You say fungi, so I'm trying to stay consistent. They surround the roots, but they do not penetrate them. And so you can see here, the white up there is an ectomycorrhizal fungi. And then the endo, of course, go in. So those are the type that penetrate the root cells. And so again, like they provide a large surface area for nutrient exchange. The big take-home message here is that this is a mutualistic relationship. What does mutualism mean? Does everybody win or does only one person win? Everybody wins. Okay? So the fungi are getting nutrition and the plants are getting nutrition. Okay? This is my fave right here, lichen. So this is, if you have test questions on this, I will especially love them. But lichens are really interesting if you are somebody that does a lot of hiking at some point in time, or maybe you live somewhere that's got relatively clean air, maybe. You may have seen lichens before. So they look and function and even reproduce like single organisms. But these are associations between a fungus and a photosynthetic partner. Typically speaking, this is a green algae or a cyanobacterium. Okay? Super important. They're very, very diverse too. You see all kinds of lichens. So in terms of the anatomy of lichens, this is one thing that's really wonderful about the structure of that hyphae, is that there's space there right below the surface where the photosynthetic partners can take up residence. So this is probably pretty predictable in what each partner gets. So as you can imagine, the photosynthetic partner is providing some of the products of photosynthesis to the fungus. And then of course, whether it's a cyanobacteria or whether it's an algae, is getting a stable place to live. No protection. There are certain things I love about lichens. I'm going to highlight two in particular for you. And then I actually have a link at the end of the presentation that gives you a little bit more on reindeer lichen. I especially like that one because it can be found in this class' favorite biome, which is the tundra. So if you look at point number 3, lichens are really interesting because since they absorb, right, they're fungi, so they're very absorptive. So they tend to grow in places where there is not a lot of pollution. And in cases where they are in environments that then become polluted, the lichens will eventually disappear. They will not do well in these places because they are absorbing everything from their environment. So they are what's called a bioindicator. The other thing is that chemical signals from the photosynthetic partner can influence the form and the shape of the fungus. So I thought that was really cool. The book talks a little bit about this idea of being able to have both in a lab independently, but really when you put them together, then the photosynthetic partner is secreting signals that actually, direct the structure and the differentiation of the fungus. So, okay. Is there anything that you love about lichens that you want to share?
I love about it.
Amazing. Okay, so in terms of spores, so one thing that if I remember correctly, I did ask on the quiz was about what are the challenges that plants face. I want to kind of stay in the same arena. When we're thinking about why do these different organisms, why did they come about? What were the adaptations that they were developing in order to meet the challenges of a changing environment? So two of the challenges of reproduction in particular faced by fungi, they needed to maintain genetic diversity. So to maintain genetic diversity, this is what you would expect. They need to find other fungi to mate with. And then also they need to be able to disperse. And so here you can see that fungi produce spores that can then be carried by the wind. And then also if insects land on them, those spores can also be taken to another location by those insects. So this should sound relatively familiar. So spores can form by meiosis as part of sexual reproduction. They can also form asexually. And then in many species, asexual spores are produced within the sporangia that form at the ends of the hyphae. So you can see that up there. Is this your red mold? This is your red mold. Yeah, this is red mold. And so because they're up and they're elevated, then they're able to be dispersed that way by wind. So here comes the question. If the goal is spore dispersal, then is elevation enough? Is elevation enough? I think you guys should think on it for a few seconds. So talk to your neighbor and think about it this way. Can you just be, if it's just up high, are the spores just going to get dispersed, or does something else need to be at play in order to permit dispersal? So see what you can brainstorm. You have like 30 seconds because we still have a couple other things we want to talk about. Yes, this. I like that. It's like jazz hands, but like fungus style jazz hands. So yes, if they're able to actually expel the spores, then the spores have a better chance of moving into a different location. Okay? So, and then we were just talking about this idea of these toadstools and essentially what their function is. And so one thing in your reading you'll notice is the book does mention a little bit about the fact that there are essentially, I think they call them pseudo-pollinators. And so, insects can come over to these because they look interesting, right? Like if I were a bug, I'd be into that. So they can pick up spores and then take them to a different location. So we're going to talk a little bit about life cycles, but we're not going to focus the way that you did on the plants. But there are certain things that we are going to highlight going through. Some of this is going to sound extremely familiar. So one thing to know is that the nuclei in the fungal hyphae are haploid. Those haploid spores are produced by mitosis, while sexual reproduction involves the fusion of the haploid cells to form a diploid zygote. So again, very familiar. It undergoes meiosis as its first division. Now here, there's a difference between the asexual cycle and sexual reproduction. The fusion of haploid cells is not immediately followed by the fusion of their nuclei. And there's differences in timing based on the type of fungus we're talking about. We're not going to harp too much on that, so don't worry. Okay, so in a few minutes, I'm going to kick it over to Dr. Brewer, but I have this slide in here because when I was reading it, all of these terms, I was like, I could see how a student would get completely turned around. So this is my guide to the crazy fungi terminology. And so what I want you to keep in mind is when you're looking at some of these figures, especially when you're reading, like, let's see, like this isn't horribly scary, right? Like that's okay, we can deal with this one. But there's some of them that as you go through, well, I won't go all the way to it. There's some of them as you go through that are, there's so many things going on that it's easy to get lost. So come back, right? Make yourself a list, something like this here, or if you want to read yourself and then paraphrase and make it so that you understand it, or when you're in your study groups and you're explaining it to each other, when you come back to those terms, then reorient yourself to the figures, okay? So one big thing here that Dr. Brewer is going to talk about in a minute are these dikaryotic fungi. And so one thing that I think is good to note here is this idea of a single cell that's got two distinct haploid nuclei. And then some of these you can even kind of see where the wording comes from. I'm not as good with my groups as you are, but for example, plasmogamy up here, you can see that that's the fusion of hyphal tips in which the nuclei are haploid. So fusion, so thinking about plasma and cytoplasm together. So hopefully that makes sense. So that's how you want to look at some of these. Right? So in terms of this life cycle, do you have things you want to highlight here?
Yeah, what I would recommend, because oftentimes you see these in isolation, but to think about it, with the fungi, it's kind of like an alternation of generations that we saw in plants, except in fungi, they can spend years and years just going through an asexual haploid cycle. And so what this will really mean is you can start with a single spore. We'll start at any point, we call this a spore. This is a single cell that is haploid. That spore lands somewhere and germinates to produce hyphae. Okay? Now those hyphae can keep on growing and eventually expand into a mycelium. And sometimes this will produce an asexual sporangium that can generate haploid spores by mitosis. So many fungi can spend years and years just doing this. Now, if they can do this, what does this imply about their environment? It implies that their environment is probably pretty stable, because if you are just going from the same haploid state to a continuous haploid state of expansion, you're not getting any genetic variability. You're just essentially cloning cells, and those cells are just expanding and creating a very big multicellular meshwork, and as Dr. Gutzler explained earlier on, this could cover miles of ground. So what this means is that the fungus isn't really having any trouble growing in its environment. So there's no really strong pressure to increase genetic diversity. What will sometimes happen is that they will then temporarily enter a diploid sexual reproduction stage. Now, how they get there varies from one type of fungus to another. So what we're looking at here is this temporary diploid state. And this state, what it looks like, is what determines how we classify different fungi. For the most part, fungi spend the vast majority of their lives in a haploid state. Some of them are making hyphae and extending them into mycelia. Some of them are just producing single-celled yeasts that can bud, so they'll bud off from each other, but they're maintaining a haploid state. There are conditions that will promote a temporary diploid sexual reproduction. For fungi, this is always just a transitory state. So compare that with our life cycle. Our life cycle is like the reverse of this, where we're always diploid, except for this tiny period of time when we create gametes, for sexual reproduction. Compare that to plants. Some plants are haploid for a lot of the time, and they create a temporary diploid state. Those we're thinking about the non-vascular plants, the bryophytes, where the gametophyte structure is the main photosynthetic structure, and the sporophyte is just this tiny little stalk. And then other plants, the sporophyte is the dominant diploid state, and then there's the temporary haploid state. So this is one way to think about fungi in terms of their reproduction. But the way they live most of the time is just cycling through a haploid state. Then they go into a temporary diploid state. How they get there, though, can vary a lot. So some of them will produce mating structures of just hyphae that can fuse with each other, whether positive or negative. They will fuse, and each of these has its own little nucleus. And then when the cells fuse, the nuclei don't quite fuse yet. That's the I say plasmogamy, plasmogamy, and should be. I've never heard anybody else pronounce the word. Karyogamy, that's what I think. You say it that way because it's similar to gametes. So karyogamy would be when these finally fuse, then you have a nucleus that's part one cell type. Part one cell type and part another. And now you've got what really is sexual reproduction. Now, typically, for us, and even for plants, what this would mean is then you would take this new diploid cell. This one is different. It's not really diploid at this point. It's the dikaryotic state, N plus N rather than 2N. This diploid state in an animal would divide and divide and divide, and you get a complex multicellular organism that's diploid throughout. But fungi don't do this. They go immediately from the diploid back to haploid meiosis. And then they can produce spores, or they can immediately start producing hyphae.
So then the follow-up question becomes, is this then
Goes
bring
right
us back
here.
over
Yep.
here?
Goes right back over here. So the distinction between the major groups of the fungi, it really all comes down to what do they do at this point? How do they go from the N plus N stage through to the diploid stage and back to the haploid stage? Because some of them do weird stuff. So some of them, like the zygomycetes, will reproduce all of these nuclei many times in one single cell. They have what's called a heterokaryon that is multiple nuclei all together. And then they'll somehow divide those up and create a temporary diploid nucleus, and then they will go back into this stage. Whereas some, like the ascomycetes and the pseudomycetes, will tend to go into this diploid state and then do something a little bit different at that point. They'll make a multiple diploid cell state and then start dividing it up into haploid cells. Those details aren't as important. So what you want to understand about the reproduction of fungi is that they spend almost all of the time in a haploid state, just growing vegetatively. What is it that will trigger sexual reproduction? It's usually some problem. It's a problem in their environment. The weather has changed. That's a big trigger for fungi. So you'll often see mushrooms sprouting up after it's rained for a few days in a row and the temperature has changed. So what's happening there is the conditions are favoring some change in their genetic material because the environment has now no longer been the same as it was before. So then they produce new spores. Because what happens is when they go through meiosis and create new haploid spores, the genetic composition is a mixture of the genetic composition of two different hyphae, two different mating types of hyphae. So this is where they get their new recombined genetic material. So sexual reproduction and asexual reproduction are two different strategies that are used by many different forms of life. Asexual reproduction is really good in that you don't have to expend many resources in finding a mate. So you don't have to worry about how am I going to fertilize two different cell types. None of that matters. All you got to do is be able to divide. The disadvantage is that you don't have any genetic diversity. you're just constantly reproducing asexually, you're cloning the same thing over and over again. And so what that means is if conditions change, you may no longer be suited for the new environment. And the change could be an environmental change in temperature, water availability, or it could be some sort of parasite or some sort of predator. So as long as the fungus can expand, 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 it will do so asexually. And as you see from some of the examples we talked about, this could happen for years. It can happen for years, that a fungus is spreading and spreading and spreading, and then something happens. Now, when these spores land, something that's interesting to me is when the spore lands, it's a single cell, and then it starts dividing and dividing and creating hyphae. And those hyphae start branching off. So the way this growth will occur is kind of like a fractal process, out from the center. The natural progress of this, if growth was equal in all directions, is that you get something like circular outgrowth from a central point. And then if conditions change, so this kind of growth is no longer favored, you go into the sexual reproductive stage. And for the different types of fungi, that means a different kind of fruiting body. So one of the groups that we'll look at is the Basidiomycetes. They're the ones that produce the toadstools, the mushroom caps that you're familiar with. When they do this, they're going to produce them usually from the areas where the hyphae are growing more actively. And that's usually on the periphery. So what that means is that they're going to pop up these fruiting bodies at the ends of the newest hyphae, which are on the ends of the rings. So when they come up, they'll come up in a circle. People used to call this a fairy circle, because people believe all kinds of crazy shit. You see something like this in the forest, and it's a circle, and that looks unnatural. Oh, witches. It was witches, or it was Satan. But no, it's just, this is where the fruiting bodies are going to pop up, because that's how the hyphae have grown out. So it's not unusual for these fruiting bodies to appear in a circular pattern because of the way the hyphae have grown from a central spore that landed somewhere and was germinated and spread over a period of time. That's not my thing.
We have 7 minutes to do them.
All right, so I'm not going to worry about mating types right now, but we're going to do this quick overview of the diversity of fungi. All right, so you have a phylogenetic tree of all of the fungi. I want you to know a little bit about each of these major groups. So first we're going to talk about the chytrids. Chytrids are mostly single-celled organisms, and they are aquatic. Now some of them will cause problems for fishes. They can grow on the fish and eat these little thick stuff, this little white fuzzy stuff. But one of the things that has caused them to become more important to us in recent years is that they can also infect amphibians. And in the last couple of decades, there's been a collapse in populations of amphibians around the world. Frogs have been dying off. And for a while, people didn't have any idea what was going on. They thought, oh, it's hormones in the water. Well, it turns out the causative agent is chytrids. These types of fungi that can infect amphibians and fish, because again, they like a moist environment, especially one where the temperature is not too hot. So they're used to growing in probably covered up forests where it's wet. The chytrids that are causing the most problems started in Africa, but the amphibians in Africa had co-evolved to deal with them so they could survive the chytrid infection. But then ecotourists would go and they'd go tramping through forests in Africa, and then they would take those same boots and go tramping through forests in South America and bring with them the chytrids that could now infect frogs that had never been exposed to something like this. And what has happened in the last couple of decades is that massive populations of frogs in South America and Central America have been completely wiped out because of this infection that was transported to them. They had no way of dealing with it. So what I want you to know about chytrids is they're bad for frogs. Zygomycetes. What characterizes the zygomycetes in terms of their reproduction is something that's more complicated than we want to know about, but they're the ones that do this kind of thing, where when two hyphae come together, they can create this really complex zygosporangium that has many, many nuclei in it. But what I want you to keep in mind about them is that they are important to us primarily because most of the moles that you see are zygomycetes. So what a mold really is usually is a bunch of these kinds of fruiting bodies, where the fruiting body is a stalk with a kind of a bulb on the end of it. And if you have a bunch of those, it looks like a black carpet, buds. So the nature of these fruiting bodies is, as Dr. Gusta was saying, it's not enough to just be high. If you can send things away, that's even better. Sometimes what will happen is, as these molds dry out, they flick the spores up into the air. And even the slightest touch of a breeze across them can cause them to be dispersed a long distance away. So when you see red molds, or you see black mold on a dry wall in a house, that's usually going to be zygomycetes. Another group we're interested in is glomeromycetes. I want you to remember these because these are the ones that form mycorrhizae. So these are the groups of fungi that are really important for plant growth. Glomeromycetes grow in the soil, and when they get in close contact with the roots of plants, oftentimes the plant root will send out chemical signals that attract the glomeromycetes to grow on or into their roots. And because fungi are really good at absorbing things, they can extend the root system of plants. They're much better at pulling water in than plants are, but they can't make their own food. That's something plants do all the time, so they just exchange materials. If you grow plants in a sterile soil environment, they will never grow as well as they would in a soil environment that has glomeromycetes. All right, so the dikarya, these two groups, Basidiomycetes and Ascomycetes, these are the ones that spend a significant amount of time in this N+N state. When two hyphae fuse, the nuclei don't immediately fuse. So when our sperm and egg come together for our fertilization, as soon as the sperm and egg come together, the nuclei fuse. That's the fertilization step. Both Basidiomycetes and Ascomycetes delay this process. So they will come together, but the two nuclei don't fuse immediately. So what distinguishes the Basidiomycetes and the Ascomycetes is the kind of fruiting bodies that they generate. So the Basidiomycetes, I want you to think of as the club fungi. They make club-like structures, and most of this is microscopic. The Ascomycetes make sac structures. Asco refers to sac. So it's really about when they go through meiosis, where do you find the spores? The Ascomycetes, the spores are initially formed inside of a sac structure. So the other things to remember about these is the Basidiomycetes, their fruiting bodies are the ones we're most familiar with, the mushrooms. The Ascomycetes make some things that are kind of like mushrooms, so some of the morels that we looked at before in a picture, and some of the puffballs, but they're also the groups that contain the most important yeasts, the ones that we use for brewing, baking, cooking. All right. So one other general thing that I want to talk about is that they can cause us problems. Fungi can cause infections. When they cause us infections, what can we do about it? Usually, fungi don't cause us massive problems. Our immune system is adapted to deal with them, and our bodies, even though we have moisture in places, the temperature and the acids that we produce often inhibit their growth. When people get fungal infections, it's usually because something has happened to change the temperature, to make the temperature cooler locally, or to neutralize acid. So people get yeast infections in parts of their bodies. It's often because the natural acid environment has been neutralized. If you get an infection and you need to get rid of it, you can't use the same drugs that we use for bacteria. Antibiotics won't do anything to fungi. But fungi are especially susceptible to sulfur-containing drugs. This is one of the reasons why lichens are so susceptible to sulfur pollution. Sulfur is really bad for fungi. So almost every antifungal drug that you can think of will have, as part of its name, azole or izole. And that in chemistry always means sulfur. So those are some interesting